Hello viewers, welcome once again to our ongoing series entitled Mendelism and Beyond. As you know, learners, we have been making extensive analysis of the cytological proof of crossing over, and thereafter we had talked about the first part of what is called as the genetical proof of crossing over, wherein we took help of tetrad analysis. Now, the first part of tetrad analysis was devoted to a situation where the tetrads were placed randomly within a sac or what we call as an ascus. But then there would be certain formulae which would govern the finding out of the recombination frequency, which would be now a starting point for finding out the genetic maps or constructing the genetic maps. Today, we have to talk about the genetical proof of crossing over in case of the tetrads if they are in an ordered form. So, whenever we are talking about the determination of gene frequency and the map distance in ordered tetrads, let me first recapitulate the earlier situation where the tetrads were not in an order and the sac was so large, so that these particular meiocytes could now have their own place within an ascus, and there were several situations which led to our calculation basis. The first idea was that if in a possibility that there is no crossing over, if there is no crossing over, then obviously the two sister chromatids, or rather all the four of them, they would remain what they were, and as a result we would get a parental ditype, which is rightly abbreviated as the PD. So, that means the entire parental combinations are present. In a second situation, there is a single crossing over. Viewers, you should view this diagram very critically, because the crossing over has taken place between, if we just put a number, between the non-sister chromatids number 2 and number 3, and therefore, this is a single crossing over. So, that means there is only one chiasma and the genetic recombination has taken place, which would now result in the formation of capital AB, capital A small b, small a, capital B and small a small b. If you look carefully, the two parental combinations are still there and the two are new. Now, this is termed the tetratype or abbreviated as capital T, capital T. There could be many such ideas which would now give rise to either the parental ditypes or the tetratypes. For example, we find there is a double crossing over in the two strands. Now, this double crossing over is a very interesting one, because it will give rise to whatever is lost in one of the exchanges is regained, or the fragment does not include the genes. So, that means we are going to get parental ditype here, and perhaps crossing over is there, but then the recombination goes unnoticed. And if there is a 3-strand double crossing over, that means it involves chromatid number 1, number 2 and number 3, as you can see on your screens, then we will obtain a tetratype, that is all 4 different. Now, we also assume that the single crossing over would result in a situation of a parental ditype or a tetratype. But then if there is a 3-strand double crossing over, that means at 2 places, then we will get a tetratype. So, ultimately we reach a conclusion that only the 4-strand double crossing over would result in all the different spores, that is the spores having all different genotypes, and this is called as the non-parental ditypes. So that means the 4-strand double crossover would yield the NPD tetrads. In other words, we reach certain basic axioms or certain basic rules. That means if there are meiotic cells with no crossovers at all, if the number is much greater than those which are showing 4-strand double crossovers, then we are talking about linked genes, because they are not prepared to recombine, they are not prepared to separate. So therefore, linkage would be indicated when we get non-parental ditypes. They are rare, or they are in a much lower frequency as compared to the parental ditype tetrads. So that means our formula would be now NPD is less than PD. So what we concluded even on the last in the last lecture was that if we have to indicate linkage, then we find that the non-parental ditypes have to be much greater in their occurrence as compared to the non-parental ditypes, and then only we will be in a position to calculate the map distance between the two genes. But the point is that these two genes should be sufficiently close, so that we have to neglect the double crossovers at the higher levels. So the formula becomes 1/2 into number of tetratypes by total number of tetrads multiplied by 100. As you can see, viewers, on the screen, we have different situations. Say for example, we have the unordered tetrads and the ordered tetrads. We find that in mushrooms, in Ascobolus and Aspergillus, we find, even in Saccharomyces for that matter, we have such large sacs that the spores, they can have their way and they can be distributed randomly. But then when we are talking about Neurospora, then we have a very narrow ascus, so the spores cannot move anywhere and they would always be present in an order. Now this is a very interesting coincidence of nature, which we have taken to our advantage to find out how the recombination frequency and the map distances can be calculated in the form of ordered tetrads. Now talking about Neurospora, we had talked about this particular wonder material even earlier, but now we can also present some more details. Now this is called as the red bread mold, belongs to the family Sordaceae and the phylum Ascomycota, and this is actually a filamentous ascomycete. It's a haploid organism and its haploid chromosome number happens to be 7. Now what a nice coincidence. The 7 chromosome number is also equivalent to the haploid 7 in case of garden pea, and the name itself, Neurospora, means that it is a nerve spore. It is a nerve spore in Greek, and it refers to the characteristic striations on the spores. That is why it is called as a nerve spore. If you remember, viewers, Beadle and Tatum were the people who received a Nobel Prize in 1958 for their pioneering work on Neurospora crassa, and it was this particular biochemical genetics work which led them to formulate one gene, one enzyme hypothesis. One gene, one enzyme hypothesis. And this simply stated that specific genes, they code for specific proteins, and you know that much later then this hypothesis was further elaborated, and people like Norman Horowitz also gave the idea of one gene, one polypeptide, and much later by Seymour Benzer as one gene, one cestron. So that means by the year 203, the entire genome of all the 7 chromosomes has also been sequenced, and we find that the sequence is about 43 megabases long, and it includes about 10,000 genes. It's a very, very important material because it has provided a rich source of knowledge for epigenetic phenomenon. Now, what are epigenetic phenomenon? When the normal sequence of genes give an alternative expression which is maybe present inside, but then it goes and gives us an alternative expression that is called as the epigenetic phenomenon. Let me also add that Neurospora shows many features which are found in higher eukaryotes and absent in both budding and fission yeasts. Now, this DNA methylation also is present here, and you should remember that distinct RNA interference is also present. So that means we have many RNAi based silencing mechanisms which are operative in the mitotic as well as the meiotic cells. So that means this is a very, very unexpected wealth yielding fungus, and we know now a lot about the gene silencing systems based on Neurospora crassa. No doubt that the fungus is nicknamed as the Drosophila of plants because of such an extensive work, and is also called as the Cinderella of genetics because it has so many mysteries to unravel. In Neurospora, what we find is that the products of a single meiosis, they can be easily recovered and examined. And actually the products are 4 in terms of spores, but these meiospores will be now called as a tetrad. We call the whole system as tetrad analysis, but then actually it is an octat, because these meiospores, once they undergo second meiosis, once they undergo the second part, they will have a second mitosis also. Now, we find that the geometry of meiosis is revealed in terms of ordered tetrads, and what is so fascinating is that the life cycle is haploid. So that means even the recessive traits will show up in the offspring, and there will be absolutely no confusion between a dominant and the recessive expression. On your screens you can find the generation of an ascus in Neurospora crassa and how the ordered tetrads they are arranged. The first meiotic division, of course, would be giving rise to a linear array of the meiotic products, which will again now undergo the second meiotic division, giving rise to 4 post meiotic products. Now, the post meiotic products would now undergo a second mitosis, which would give rise to these sexual spores, which are the ascospores, which are around the nuclei, and they will be now 8. Actually, these 8 are the duplicate products of the 4s. We find on our screens another beautiful diagram of the ordered tetrads, how the genetically identical cells could be got in an octat. And the products of meiosis are actually 4, but then we have the replicates of 4 of them. So, we have the 4 spore pairs. So, the ascus should be called more of an octad, or if you still like the term tetrad, so it would be what is called as the tetrad pairs. That is, it is equivalent to 4 products of meiosis. On your screens you can see a detailed view of the life cycle of Neurospora and how the conidia are formed and how the dikaryoticization takes place when we have the fusion and then finally the occurrence of meiosis. The ascospores are actually located within the perithecium or the fruiting body, and from there these perithecia, then they are the walls get ruptured and the spores, they are released. The point here is that the spores will not be allowed to be released. One can remove the ascus and then remove the ascospores one by one in the same order in which they are arranged in nature in an ascus. That would make a very good study, and that means the morphological traits like color, pattern of growth and many other features can be visually seen and then later genetically analyzed. Let us take now certain genetical situations as they occur in Neurospora crassa. For example, there is a cross between a normal, that is A plus, and a mutant strain, which is A. Now, this would give rise to a linear arrangement of 4 normal spores, which is A plus at one end, and this would be followed by 4 mutant spores at the other end. So, that means we have a basically what is called as a 4+4 ratio. Remember viewers, this 4+4 was a different one. That means actually it was 2+2. Now, we find that such an arrangement can be disturbed provided crossing over takes place between the centromere and the gene, because we know that crossing over will take place at 4 strand stage and not at 2 strand stage, and this is going to produce a different ratio altogether. This would be 1:1:1:1, which would later be duplicated as 2:2:2:2 ratio. So, we have now 2 different situations. Let us take a concrete example here with the help of certain figures, how the pattern emerges in 4+4 and how does it deviate in terms of 2+2+2+2. So, suppose we took mating type A and we cross it with a type small a. The zygote after fertilization would be having capital A and small a. Now, there are a whole lot of possibilities. We call the first possibility as the first division segregation. What does it mean? It means that there is no crossing over between the mating type locus and the centromere at all. Now, in this type of situation, it is natural that the alleles capital A and small a, they will segregate to their different nuclear areas just after the first meiotic division, and then it would be a replication. On your screens, you can find a situation where there is no crossing over at all. So, the chromosomes they come together, they do not cross, and then later the centromere splits and the chromosomes become double, and as a result we can have a situation of 4+4. So, either it is 4 capital A followed by 4 small a, or it could be the reverse, that is 4 small a followed by 4 capital A. It means one and the same thing. That means no crossing over has taken place at all. If we were to look at this diagram a little more carefully and in details, the first division segregation, which is abbreviated as FDS and which does not show any crossing over at all, would have a typical 2+2 arrangement. And as I said before, it gets replicated and becomes 4:4. Now, this situation in an actual ascus, if we find with the help of genes, as I said before, the capital A could come first followed by the 4 small a's, or vice versa. In both the cases, the arrangement still remains 4:4, which is the original 2+2. If another situation is got, that is when crossing over is actually taking place, this will be called as second division segregation, and that means there is a crossover between the gene and the centromere. Now, this is going to result in 8 ascospores, no doubt, but their arrangement is different. Their arrangement would be 2 plus 2 plus 2 plus 2 pattern. A scientist by the name Lindegren, he studied the inheritance of 2 pairs of characteristics in Neurospora, and he was the one who presented the first evidence to prove that crossing over occurs at 4 strand stage, and it involves only 2 of the 4 strands. Now, we will see a little later which ones are those 2 strands that at any one place that they are going to have crossover. Let us look at this diagram, which suggests that if there is a single crossing over, and it will prove that it has actually taken place at 4 strand stage, because the zygote is formed, and then the zygote, the homologous chromosomes would now they have a replication, and if they had a replication before they crossed over, then it is the right situation. As you can see on your screens, when at the time of ascospore formation, there is a single crossover, and we have 2 parental combinations and 2 recombinants, which is actual case even in general meiosis as well as reflected in the form of the tetrads. And we can have a testimony to this, we can prove this in the case of ordered tetrads. So, I would repeat that the second division segregation, which is abbreviated as SDS, is going to involve 1 crossover, and it would result in 4 different types of genotypes in the ascospores, which later duplicates into 2 plus 2 plus 2 plus 2 pattern. So, that means this is a deviation from 4:4. So, this deviation, which is a result of crossing over, the percentage of the second division segregation SI are actually used to calculate the map distance between the centromere and the genes. Now, when we were talking about 2 plus 2 plus 2, that is the second division, we have 4 ways how to make oneself understand that how the second division segregation takes place. Let us look at a situation where we have, if you see carefully on your screens the color of the ascospores, it happens to be in first 2 plus 2 plus 2 plus 2, in the second it is 2 plus 4 plus 2, but the 2 whites are on the extremes. The third situation happens to be again 2:4:2 and not 4 plus 4, as was the case in first division segregation, but here the darker ones are at the edges, and we have a situation which is reverse, that is in the fourth we have the reverse situation of the first one, where 2 whites, 2 black, 2 whites and 2 black, which means 2:2:2:2. Now, this would depend which of the chromatids went into crossing over. Let us look at a situation. If you can see the 4 chromatids of these 2 chromosomes, let us first name them from top 1, 2, 3 and 4. Now, this second division segregation is between the non-sister chromatid number 2 and 3, and this is going to result in a pattern which is 2:2:2:2:2. This is again a normal second division segregation. There could be another aspect, because we know that crossing over is going to take place between 2 non-sister chromatids. So, by that account 1 and 4 are also non-sister. So, if crossing over takes place between 1 and 4, again we get the same result, which is 2:2:2:2:2. But then, if we were to have a situation where crossing over takes place between the number 2 and number 4, now this is going to give rise to what is called as 2:4:2 pattern. It is second division segregation, all right, but then the arrangement and the geometry of meiosis of in terms of the ascospores becomes different. And lastly, if the crossing over were to take place between 1 and 3, they are also non-sister chromatids. Again, we would have a situation of 2:4:2. What would be the difference? As I said in the beginning, in 1, the 2 darker ones would be at the periphery and the 4 lighter ones in the center, and in the other situation of 2:4:2, they would be in an opposite orientation. So, finally, in the case of an ordered tetrad, we find that the ascus is so tight, that means they are not going to move randomly, the ascospores, and the map distance will be calculated by the formula half number of second division segregation of the SI divided by total number of SI. That means the first division segregation and second division segregation taken together multiplied by 100. So, that would be the basis of the calculation of the map distances in an ordered tetrads. In some of our next lectures, we may take up the numericals for this particular purpose. Thank you.